Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. Lara Campana is the Senior Vice President of Research and Translational Sciences and a co-founder of Resolution Therapeutics, a biotech company developing novel regenerative macrophage therapies for inflammatory and fibrotic diseases. In this episode, Lara and I talked about her fascination with macrophages, the origins of Resolution, letting go as a founder, and a mindset shift that's needed to move from academic research to pragmatic, patient-focused drug development. I'm back on Careers in Discovery, and I'm delighted to be with Lara Campana of Resolution Therapeutics. Lara, welcome to the show.
Thank you very much. Thanks for having me. It's a pleasure to be here today. Looking forward to our conversation.
Great to see you. And Lara, you and I have spoken, many times about various different things relating to Resolution and Biotech and all sorts of stuff. But it's a very, very exciting time for you guys at the moment. And I'm fascinated to learn a little bit more to start with about regenerative macrophage therapy, how you're leveraging that at Resolution, and where you're at with the company today.
Absolutely. One of my favorite topics is at Resolution, we develop regenerative macrophage therapy for inflammatory and fibrotic diseases. Mm-hmm. Our aim is to leverage the antifibrotic and anti-inflammatory characteristics of these cells to drive tissue regeneration and clinical improvements. Our first target indication is end-stage liver disease. People sometimes may be surprised to hear that, late-stage cirrhosis is an inflammatory and fibrotic disease, but this is exactly what is a liver that for various reasons has been damaged chronically over the year, has progressively developed a scar, which is what in jargon we call fibrosis, has developed cell death and cell senescence, and as a result of it has developed a chronic level of inflammation. So it is really a really great target for regenerative macrophage therapy, because by acting on both the pathogenetic mechanisms, we are able to drive liver regeneration and then down the line improvement in the clinical outcomes of these patients.
Yes.
And I think it's worth noting that these patients today have literally nothing in terms of medical therapies. Mm-hmm. In the worst cases, they can access transplant. But you don't need to be a doctor to know that not everyone gets a match when they're waiting for an organ. Liver transplant is a major surgery, and these patients oftentimes have issues with coagulation and other issues that do not allow them to withstand such an invasive surgery. And then there is an aftercare that is a lifelong immunosuppression. So it's not without its own limitations.
A lot of people die while they're waiting for the transplant as well, right?
100%. Unfortunately, that's true. That's true in different proportion in different countries. We were recently learning that, for example, in Spain, they're particularly good at moving people through the list. Whilst in other countries, other Western countries, unfortunately, that's really true. Up to 20% of people, unfortunately, die while waiting for an organ. So as a major unmet need, unfortunately, is growing. If you look at statistics in the UK for liver disease as compared to other chronic diseases like cancer or heart diseases, cardiovascular diseases, liver disease is the only one that's been steadily increasing in the last 50 years. So is a growing population, growing unmet need, and our cells seems to be the perfect fit to solve the problem. So we're very excited. We are starting literally as we speak our interventional study with our engineered regenerative macrophages. So watch this space.
Fantastic. And so going sort of one level below that, then why macrophage regeneration? Like why that approach, and what's the sort of benefit of that?
Yeah, so again, I think it sometimes can be surprising, like you're trying to heal the liver or the lung or another organ, and you're like, well, use lung cell, you'll use liver cells, so surely that's the best way of doing it.
Mm.
And actually, this thinking is slightly flawed by the fact that in our organisms, every time you have a chronic damage, you also have this chronic inflammation and scarring.
Right.
And chronic inflammation and scarring prevents the local cells from regenerating. So your liver is good at regenerating unless for many years you damaged it, and then all of a sudden there's too much inflammation and fibrosis for this liver to regenerate. And therefore it doesn't create enough mass that then does the liver job.
Yes.
So what in simple terms, the reason why you do regenerative macrophage therapy as opposed to specific organ cell therapy is to condition the environment to switch from hostile to regeneration to permissive to regeneration.
Yes.
So in a way, you're teaching your organ to get down a notch, calm it down, and allow the local cells to regenerate and then do the job. And actually, it's been shown in the literature that if you inject the organ cells into the wrong environment, they will be led astray and they will do not their job properly. Yes. So, it's like one bad apple in a box of apples, and then all the apples go bad, right? And it's a bit like the same idea. If you put your cells in an environment that's not correct, that's not permissive to regeneration, your cells will go senescent, your cells will die. So we act at the underlying issue to then allow native regeneration to happen.
Isn't that interesting? I talk about that quite a lot from a people and culture point of view. And it's so interesting to hear that happens at a cellular level as well.
I always find it fascinating how sometimes biology mimics human behavior. Yeah. And if you think of it, human behavior is biology. Absolutely. So maybe it's not by chance.
No.
But yeah, that's true. It's true.
Very interesting. And so you are sort of 5 years into this journey with Resolution. You were one of the co-founders. You run the research and translational science groups there. And I believe it's a very exciting time at the moment.
It is. So currently we have open observational study, which we almost completely recruited for. Mm-hmm. And again, one of the key unmet needs in the field is knowing about this patient population. If a patient population doesn't have a medicine developed for them, usually you don't know much about them. Yes. So our observational study really allowed us to learn better and more into the details about the natural history of this disease. And we're also collecting blood samples to do further analysis to understand the landscape of these patients. And very recently, we got approval in both UK and Spain to start our interventional studies. So the study in which we are going to test our medicine, and we have just started recruited literally as we speak. So hopefully we will have our first patient treated soon. And that is a hugely not only exciting, but also rewarding. I always say that at the end of the day, I'm in this business to make an impact, to change for the better patients' lives, that patients that before didn't have an option, and now all of a sudden they have hope for something. So hugely rewarding, beside exciting.
Absolutely. And to have something that you've worked on for 5 years and, well, more than that, to get to that point is very, very exciting, and probably a little bit terrifying, which is probably a good mixture.
Yes. Is as we were discussing, is a great mixture of adrenaline, hope, and holding your breath really, really tight to see what happens. There's always that element, obviously. I think it's very important never to lose sight that the people at the other side of your job are humans.
Yes.
And therefore, you really want to do the best for them, and therefore, you always hold your breath up until everything has gone well, all the boxes have been checked, and you know that person is on a better path. You will always hold your breath a little bit, and I think it's healthy to do it. I think otherwise you just slip into this very detached, dry kind of attitude, and I don't think that serves you particularly well as a medicine developer. You remain an R&D research person, and you do not transition into that medicine development mission that ultimately your key stakeholder is your patient at the end of the day.
Yeah. Talk a bit more about that, because I think, we'll talk about your career and your journey through it. But obviously, you had a reasonably long academic career prior to the founding of Resolution. And this is perhaps a stereotype, but the stereotype would be that academics work more in theory than in practice. And that R&D versus medicines development is a really interesting dynamic that often requires a bit of a perspective shift. Talk about that sort of journey as a scientist that you've been on towards medicines development.
Yeah, absolutely. So I started studying medical biotechnology at the San Raffaele Scientific Institute in Milan. It was a very forward-thinking course for being a course that started in 2003.
Yes.
So, laptop weren't a thing back then. And
I remember,
So day one of your course, you received a laptop that you would keep for the 3 years and would allow you to get that IT literacy that you need, but also enter into the idea that as a person that does research, you cannot be only doing research.
Right.
And our course was within a medical school. So for a lot of the topics, we had the same professor that was teaching to our clinical counterparts. And when they were spending time learning in the wards, we were in the laboratory learning from actual scientists about science and about research. And again, incredibly forward thinking. We also had on the third year some courses on health economics, IP. So it kind of started to seed the idea that for how much, I went into it because I was passionate about science, right? And that was my drive. And that is the first time that something clicked for me, that what I really wanted, what really made me excited was to get into somehow the kind of more clinical facing translational aspect of research, that there was more than just studying what amino acid interact with what other amino acid. There were people in my class that were very, very much orientated on that side. For me, those years were very formative. They also exposed me to a lot of concept of cell and gene therapy, again, very early on. But the fact is that what eventually became LibMeldy, LenMeldy, came from research developed in San Raffaele. Some of those developers were our teachers. So we were exposed very early on to these concepts of cell and gene therapy. And I clearly remember being blown away. There were these boys that before were in these bubbles, they couldn't do anything. And then all of a sudden they were playing football with their friends. And I really remember thinking, wow, this is something I want to be part of. So I suppose that there was always a little bit of a seed in me that didn't quite accept just being in your ivory tower and doing your research. And by the way, this is really important, right? Really important people that do basic research or fundamental research, because that's the knowledge we leverage to translate. But that wasn't my call, I suppose, from early on. So also as a laboratory, I chose a laboratory that had quite a translational kind of flavor. We did lots of quite cutting edge stuff. I was doing MRI in mice on the muscle of mice to follow regeneration in a way that could be clinically relevant. And then eventually they developed a methodology to follow inflammation in the muscle in patients in mice with autoimmune diseases, polymyositis that affected the muscle. I met macrophages early on. And I married them. My husband knows that he has to share a part of his heart, of my heart, with the macrophages. So I suppose I always had that idea, okay, these cells are really fascinating. They have a huge potential to heal tissues. And I'm really interested in how do you make an impact on patients. So those have always been my North Star as I progressed in my career. That also led my choice of moving to Edinburgh, where they started to have a preclinical program testing these cells in the liver. Prompted me to study more also in acute models, thanks to collaboration with one other postdoc in the laboratory, Professor Forbes. So yeah, I suppose I was never probably a classical academic in that I always had that kind of orientation towards transforming into something impactful. The biggest mindset shift, I suppose, that I went through when I moved to Resolution is learning what I call pragmatic science.
Okay.
A lot of my team come from academia.
Yes.
Because we are the research and translational science team.
Yes.
So sometimes it's easy to slip into the trap of, oh, this is also interesting, and this is also interesting, and this is also interesting. And the reality of it is in biotech, you have draconian timelines on relatively small budgets.
Right.
And therefore, you need to be ruthless in your prioritization. And that's what I call pragmatic science. Your science still needs to be solid, robust, needs to withstand the test, the scrutiny of external KOL, external bodies, peer reviewing, but needs to be pragmatic. If you start spreading yourself in your direction, which is something that in the academic world you have the gift to do, even if you are translationally orientated, you do not have this gift in biotech. And therefore, that's probably the biggest mindset shift, I would say, that happened. It clicked very early on that to be the case to succeed.
Yeah, and I suppose it's those interesting questions that you have to ask, right? Around why are we going to do this experiment? How does it help us move towards our goals? Do we actually need to do it? And I think, there's sort of always going to be that drive to understand the disease more or understand the target more or that kind of thing. And to a point, that's really important. There comes a point where there's diminishing return, right? And understanding it a little bit more doesn't actually get you closer to what you're trying to achieve.
Yeah, and also sometimes I tell people, we need to keep notes of this thinking because that can help in the future. So, during our Series A, our goal was to get to CTA in the UK and Spain. I was also in this phase, I was also the asset program lead, so it was a matrix team. So I was also leading the kind of coordination of the various teams within the company to drive the development towards CTA approval. And I always tell people there are few things that we need to do in the life cycle of the development of this drug, but we cannot do it now. Because if we use these resources now, then what happens is that we do not have money and time to do, say, our GLP toxicology study. And unfortunately, there is no a day in the week in which a regulator will give you a green light for a phase 1-2 study without a proper GLP toxicology study. So we have small team, a reasonable budget, but not an extensive budget. So we need to be pragmatic. Do we want to do more pharmacology? Of course we do. We can do it later. In Series B, as we prepare for pivotal, which is what we are doing now, we're going to kind of fill in the blanks, if you will, that we left, but we successfully delivered our CTA filing on time and our approval as well. So I think that's what I call pragmatic science, and I think my team is sick to hear about it. But hopefully that means that we can still do robust science, but in a directional way.
Yeah, absolutely. No, it's very interesting, and we talked about this a little bit when we were talking about the work you're doing at Resolution, but you lit up when you started talking about macrophages, Lara. So is it just the ubiquity of them in the body? Is it the regenerative, like the therapeutic potential? What was it that kind of made you fall in love with macrophages?
So we were having a lecture on what were the traditional functions of macrophages, like phagocytosis of bacteria,
Yeah.
sterilization of the tissue, etc. And I clearly remember a lecturer saying at the very end, saying, "Oh, in the deck, I leave you a link to this new review that has just been published because it's emerging that macrophages may also have an active role in regeneration."
Okay.
Right. And that kind of something clicked. And I went, I read that. She said that it wasn't part of the exam or anything. And obviously
Yeah.
I ignored what she talked
Yeah,
about
yeah,
before,
yeah.
and I went straight for it because I found it so fascinating. And what clicked for me is that the immune system is itinerant. It travels to your body, goes everywhere. The precursor of macrophages are monocytes, are in your blood. They go everywhere. And they're very clever because they understand when there is damage in a tissue. Because the tissue produces molecules that are like little flags that ask for help, are like the Batman sign that calls for help, right? And the monocytes come in, they become macrophages. And they have this incredible ability when you have a damage that is self-limiting, like, your child slips and falls at the play park, scratches their knee. Macrophages go there, monocytes go there, they become macrophages. They start clearing the mess. So they
Yes.
start sterilizing the wound. But what they do is that those actions turn them into what we call a regenerative phenotype. And what I like to imagine them like is like orchestra conductors. And they're really good at calling upon different molecules and different cell types at the right time, at the right magnitude, for the right amount of time. And they create this beautiful symphony that then allows the tissue to heal. And I found that fascinating because that works beautifully when we have an acute damage.
Right.
And is completely derailed when we have a chronic damage.
I see. Okay.
Because when we have a chronic damage, what happens is that we have this simmering inflammation that is almost like a soup that bubbles in the background while you're doing all your other activities. It's on the stove and keeps bubbling and bubbling away and never resolves.
Right.
There's never this initial spike of activity that then leads to this second spike of regeneration. There's a continuous vicious cycle of a bit of damage, a bit of regeneration, a bit of damage, but never really closes.
Yes.
And what fascinated me at the beginning was like, how do you understand what happens in the self-resolving situation? And then you teach the chronic situation to behave the same, right? How do you teach your chronic organs, the chronically damaged organs, to behave like an acutely damaged organ? And the way to do it is through your immune cells, because those are the one that coordinates the process. So for me, that was the drive, and I suspect what also fascinates me is the fact that it's so ubiquitous, as you were saying, and therefore crack it in the liver, that maybe you can crack it in the lung, you can crack it in the skin. You then open up a plethora of diseases that have underlying similar pathobiological mechanisms that then can benefit from regenerative macrophage therapy. So it's been a long-standing interest of mine and real passion from early on when I first heard about it.
Yes. Yeah. Interesting. So it's kind of like, if you can harness it, I suppose, having a crack team of paramedics that you can send to tackle anything. And okay, interesting. So tell me about then where the resolution concept came from, because you were in Edinburgh by this point. You were a postdoc there, and you, I know you still have an affiliation with the university. There's normally kind of a slow bubbling of conversations, and then there's a point at which someone goes, okay, let's do this thing. Tell me a bit about that origin of this.
Yeah, so I suppose one of the things that need to be recognized is that in Edinburgh, Professor Forbes had the courage for the first time to try these cells in a human being with liver cirrhosis. And that generated the phase one study that was a 3x3, 3 patients, 3 doses, dose escalation. They showed these cells were very well tolerated. There was no control arm, so it was very difficult to say whether there was an efficacy, but some biomarkers showed some movement in the right direction. The patients seemed to be doing well, and those are patients that normally go downhill relatively quickly. So that inspired in a certain amount of hope that this could really work.
Yes. And
also sparked the interest of Syncona, who's our current main backer and funder. And they started to make contact with Professor Forbes. There was a bit of to and fro. We were never quite convinced that the technology was mature enough and that was strong enough. In the meantime, Professor Forbes started a phase 2 study after the phase 1. This was a proper randomized controlled study, and around that time in 2017, between 2017 and 2018. That's when we decided that this thing could have been a go.
Yes.
And but what we assessed is that probably the technology was a little bit immature. So I remember Stuart in a corridor saying, "Look, we're ready to go. 1st of March 2018. If you're in, I need someone that develops the technology further. And there's going to be 2 more postdocs coming on board. What you think?" And, that's one of those conversations that happens to you once in a lifetime, right?
Yes.
You've been studying these cells for years. You've been working towards something like this for years. I've done medical biotechnology, always been interested on the business side as well. Done lots of teaching, so became a big fan also of team culture, team building, and all of that. So for me, it was just the perfect culmination of all of that. I was like, okay, I'm in.
Yes.
And I just didn't even have to think about it. At the time, I was applying to fellowship to start my own laboratory. And I was just like, no, I'm in. I stayed 2 more years as a postdoc as we were developing the technology. So that was kind of hybrid funding, sort of, maybe you can call it seed, probably, type of thing. And I was fully affiliated with the university still. And then in 2020, we spun out with Stuart and the 2 other postdocs that then joined the project. And I was incredible. Trying to do it in the pandemic where everyone was scattered, and we had to postpone the actual spin-out date because obviously we wanted to be sure we could operate, because you don't want to start a business and then be idle for months and months. So the initial date was April 2020, which clearly didn't happen. And then we ended up end of August 2020 and operational from the 1st of December 2020. And it's been an emotional roller coaster ever since, but incredibly rewarding.
I'm sure. And so you started out, as you said, in this very hands-on position of you and 2 other postdocs. Let's get this technology working. Let's develop it. Let's move it forward. And of course, then the journey over the last 5 years has been sort of increasing responsibility, getting more involved in the running of the business overall, running groups rather than running, studies and kind of advancing things. There's a lot to unpack there, of course, and I'm asking you to summarize 5 years of your life. But tell me a bit about that journey from director of biology to senior vice president, and the sort of the career journey that you've been on over the last 5 years.
So as director of biology, I had my macrophage biology unit. Initially, I had 2 scientists, but very early on in the biotech journey, I realized that it's very unpredictable because for various reasons, one of the postdocs that was a co-founder decided to come back to his nation of origin. He had to lead a group of 4. So out of 8 scientists in the company, all of a sudden I was line managing 6,
Right,
and
I
having to set the strategic direction of the science, starting the company, kitting out the laboratory, my other fellow postdoc that transitioned to director role. And I suppose that taught me very quickly that you need to be very good at prioritizing. And what I was doing at the time, I was leading both the cell engineering unit and the macrophage biology unit.
Yes.
And at the time, we also had a small group of 2 scientists that were trying their hand at iPS-derived macrophages, which is then a project we eventually closed and didn't follow up on. And at the time, my main responsibilities were to set the a kind of strategy to screen various payloads to find out which one were the most suitable to improve the anti-inflammatory and anti-fibrotic characteristics of these cells, to lead the setup of a mode of action based assay suite that could allow us to do this screening, but also to verify that our engineering was good enough to do the screening. That had to happen also relatively quickly because we were expected to come up with at least one candidate per category by September, starting in January with so our first scientist walked through the door on the 5th of January 2021. So I also very quickly realized that if you do not take care of your team and deep care of your team, and you do not set the tone for how you want to work as a company, then your impact immediately decreases and you end up in a whole lot of troubles and a whole world of pain, because the team does not only need to trust you. In biotech, you need a team that's willing to go the extra mile.
Of course.
For the common mission, not for you as a person, but for the common mission that you represent. So for the common goal that we all have, and there is a clear direction that we share, and there is a clear goal that we're all trying to achieve together. And I must say, some of my other colleagues that were on board at the time, particularly our CEO at the time, someone else that was coming from Syncona helping us, they all had a huge amount of influence in me understanding this and accompanying me in understanding this, and also kind of really transforming my outlook from I just care about the science to I care about the holistic view of leading these teams, even the team that I wasn't bargaining for having, towards success and towards delivering what are our key milestones, our key deliverables. In July that year, a proper director of cell engineering joined, which also taught me a very good lesson, which is at the beginning, this biotech journey is all consuming. I don't think I had an hour off between December 2020 and July 21. But if you wanted to raise your practice and grow as a professional to your next level, you need the brain space. You need the time to reflect on your gaps, to reflect on your development journey, and to critically the development journey of your team. Because if you develop but your team stops developing, that's like the whole company stops developing. So you need the brain space to think about that aspect, to keep upskilling your team, so that you become, your company will go on a trajectory, will mature towards having a medicine in the clinic, and you need to mature with your company. Otherwise, you hold your company behind, you hold your deliverables, your goals behind. So that was a very good lesson, because I had more free time and all of a sudden I could have more impact, and I was better at delegating, and I was better at allowing other people to grow in their own space and to become their own person. And I also learned that this is incredibly rewarding. So for me now, one of the key aspects is keep having my team growing, and every time they have a milestone, they deliver the presentation by themselves. They don't need my supervision to do something. That's a huge moment, and I think that's what I learned quite early on in the journey, something that really brings a lot of fulfillment to me, the kind of human people aspect. The culture, culture is what everyone uses, but that aspect. And then I became in 2022, I became a VP of macrophage biology. And that was really a transition because I was taking more and more of the matrix leadership as well in terms of asset lead for RTX-001, which is our lead regenerative macrophage candidate. And again, that taught me that I needed to get a lot better in my influencing skills, and the ability to influence without managing, and the ability of setting expectations without having the direct power or enforcing expectations, and also taught me a good lesson on do not assume that everyone sees the world the way you see it, right?
Yes.
When you are the asset lead, and oftentimes you are one of the very few women in the room at the beginning, that got better as we grew bigger, obviously, but the beginning that was quite a solitary position. One of the probably the youngest, or one of the youngest in the room, may trigger some people's biases and beliefs in ways that you do not think should happen, but does happen. And you have two ways of reacting to it. You can kick and scream about it and be the victim, or you can decide, okay, I'm going to fight this, and I'm going to show that I can do the job, and I'm entirely qualified to be in this position and do what I'm doing. And that's always... I kick and scream sometimes, and I moan a lot, probably. But my preferred way is certainly, well, let's show I can do the job. That the best way to answer all of that is showing that you can do the job. And I suppose one of my main achievements at the time was really us getting to nominate a product that we could then put into proper process development. It was another big growth phase of the company. So I participated in a lot of interview and recruited a very high performing team that led us to achieve all the goals that we had to achieve. We had a comment once from one of our consultants that told us, "You're the only biotech that do what they say by the time that they say they're doing it."
Yes.
And I take it as a very big vote of confidence for the team that we built, and I contributed to build in that period. And for me, it was a very big, again, learning point, right? Like, how do you? Most of it was gut feeling, probably, because I couldn't say, I wouldn't say that I was very structured in my thinking at the time, but instinctively I knew that the more diverse your team is, the better you will achieve your goals, right? Because if you only hire people that look like you and behave like you, you're just filling a lot of already filled spaces, and you're left with a lot of blind spots.
Yes.
And a team that sometimes disagrees and that has robust exchanges is actually a gift, and that's probably what that part of my growth taught me, for sure. And we do have a very, very diverse team, of which I'm really proud of our Resolution. And I must say our current CEO is also super supportive of this approach. And it really sets the tone for how you lead the organization. And then, like, my last step was to just take over the department, become VP of Research Operations, and then move into the SVP of Science, Research and Translational Science. And that was really the time in which I started to coordinate more senior leaders, that all of them had their group underneath them. One of my most proud moments was that as I left the role of VP of Macrophage Biology, the person that became Director of Macrophage Biology was someone they grew with Resolution from laboratory scientist to that level. Incredibly fulfilling. Great to see that was the case. She's amazing. And I think this was again a really good demonstration of the culture of growth that we tried to woven into the organization.
Yes.
And I suppose that taught me some few lessons around, one of the people that initially I line manage was someone with a huge experience in a very different field from mine. And something just clicked for me saying, that's such, it may seem hard to you at the moment because there are lots of gelling, there's lots of gelling that needs to happen, but what a gift that you can be exposed to that, you can learn, you have things to offer, and
Yes.
they have things to offer you, and that dual exchange is actually what you should aim for with all your senior leaders in your research department. And that's really changed the way I then related progressively with also the other people, starting to have more to and fro, and really starting to establish that two-way kind of directional growth, right? Where everyone growth in the process, you as a leader and them as leaders themselves.
Yes.
So, and obviously hugely satisfactory, because that was the time in which, as a matrix leader, I led the team to CTA filing and then CTA approval.
Of course. Yes.
And that taught me another lesson, which is it's good to let go.
Okay.
But, I did mine. I did drive the team as successfully as I could to CTA filing. Now there's someone else that took over the matrix team.
Right.
And that's the right thing to do, because then what I'm asked to do is to look what's the next big thing for Resolution.
Of course, yes.
Like put my innovative and kind of processy brain at the service of something that's of value for the business, which is what's the next big thing.
Yes.
Obviously, we will still work on our lead asset.
Of course.
Because we have all the kind of translational science part, there is all the regulatory part, preclinical regulatory part to support the following phase 3 studies, pivotal studies, etc. But certainly there is now a big part of my brain that's now free to think about what's the next big impact we can have. Yeah. And, we've recently identified we want to go into lung fibrosis and chronic graft versus host disease and acute graft versus host disease, which is this condition that people with allogeneic bone marrow transplant develop. And hugely rewarding, thinking now I have the opportunity to expand the impact of RMT beyond the liver. So, so that was the lesson, I suppose. That it's good to let go, to make way for bigger, better thing, even if you have that kind of emotional link, because that's, there's your name on that patent, and you send off the investigator brochure that say that combination of payload is the one that's gonna do the magic, and it's gonna be well tolerated by the patient. So that there is that. It's more like when I always equate that when my nephew came and visit me, and then I had to have him flying back by himself to Italy. And I was like, I should have been on the plane with him. What a terrible auntie I am. But actually, it's a growth opportunity for him.
Of course, yeah.
And gives me more free time. So that is like
it's a good analogy.
So it's a win-win.
Yes, but it's hard to let go sometimes. As you say, your name's on it, but also you've worked on it for 5 years, you've nurtured it, you've grown it, you are kind of, you've been responsible for it for all that time. It's hard. There's a couple of things you talked about there that I thought were really interesting. This idea of the brain space, the thinking time, is really key. I think it's amazing what even taking an hour a week to just, without much of an agenda, to just think about where are we at, what are the issues, what's the reason for them. Like, until you do it, it's really easy to miss the fact that you're not thinking that much about what you're doing.
Yeah, that's absolutely true. I try to be quite deliberate about that in terms of having certainly an hour a month in which I have a bit of structured thinking around key questions.
Yes.
You probably know the answer to inside yourself, but you do not focus on them up until you do not ask. It's a kind of journaling style type of thing. And then I do try quite this is gonna sound really lame, but one things I really love doing is on Saturday morning I go for my fitness training, which is quite intense. I come home, have my lunch, and then I love spending a couple of hours on the couch with a cup of tea, doing some mindless scribbling and just literally like with my blanket, like just being there and just be in the space and just have the gift of time to say what are the things I care about, what are the things that are bothering me, why do I feel that this isn't, why is this not sitting right with me, that should sit right with me, why it doesn't, and then maybe there's something that comes to you saying, oh, that's why that doesn't sit right to you. Now it can sit really right for you if you solve this one thing, and that is what you really need to care about. So next week, try and care about this, or next month, or you do not have time now, but make a note of it, and then work from there, and make sure that you resolve that issue or that question. And I think learning that this is not wasted time for someone that's a laboratory scientist. I'm very much an initiator. I quite like leading the way and starting things. It was quite counterintuitive at the beginning. I did feel sometimes I was like, right, I'm being basically lazy, staying an hour or 2 hours on the couch doing nothing. And actually then when you see the impact of it, my team sometimes jokes and it's like, oh, Lara, you've been thinking, because maybe I come to them with something.
Yes. And I think there's that real power in, because when we're in the midst of stuff, we focus on, oh, this is the challenge we're facing, or this is the problem we've got, or this is the issue. But just taking that step and going, yes, that is the issue, but not how are we going to solve it, but why do we have that problem, or what's causing that, or why are we where we are? Often will show you that the issues that you face are often the symptoms, not the actual underlying cause, right? And there's real power in that. There's something else you talked about that I think is probably a scenario that a lot of people find themselves in if they're on this sort of biotech journey, is that, you found yourself as the youngest person around the table, you found yourself as the only woman around the table, but not only that, you had transitioned from an academic environment where the people who were running the company were the PIs, the professors, so there was already that sort of learned hierarchy even before the company began. Just interested in any thoughts you had on, you talked about backing yourself and showing you could do the job and kicking and screaming when it's appropriate. Sometimes it's worth doing. Any other thoughts on sort of overcoming that? Because I think sometimes it can be a limitation that people put on themselves, that the people at the top of that hierarchy aren't imposing the hierarchy, but we don't push against the kind of implicit hierarchy that's there.
Yeah, absolutely. I think, so for me, there's a few things that come to mind. And if you do have good mentors and good sponsors and good people around you, I think it's actually good to speak about it. It's good to speak about the difficulties you find. It's good to speak about how you felt in that situation, or how that made you feel empowered or disempowered, and why. And generally speaking, one things I've learned is that there's a lot more people that care about you developing than the one that do not.
Right.
And the reality is that if you do develop as a founder, as a founding scientist, the company develops a lot better,
100%.
A lot quicker, a lot faster. And people do want you to develop. So if you do have those thoughts, find the person or people you trust and make sure that they help you unpack that. And sometimes it's good to have a coach, right? I worked a lot with a coach. They gave me also sometimes some language tools to come out of certain situation better. I clearly remember once they told me, like, they cannot challenge me, and they're like, well, in this situation you didn't behave like a scientist. And I was like, what do you mean? Outraged that they even suggested that. And they were like, well, you were not curious, you didn't ask any question, you froze in that situation thinking that they were attacking your practice or your thinking, but you didn't ask any question. Right. Next time you're in that question, ask them, can I ask you please to clarify this thing? Or please, can you actually articulate what is unclear to you, or what doesn't inspire trust in this approach, so that we can address it, right?
Yes.
And I think, again, working with a coach, there's a kind of a dry vision, because they're not part of the business. They don't even understand what you're doing. You don't speak about the specifics of your science. You just present the scenarios. I think has a lot of power as well. So don't. Yes. I suppose that trust the people around you and find support, but also find someone that can look at you from outside and can give you that license to backing yourself up and give you the tools to back yourself up.
Yeah, absolutely. No, it's a really powerful message, and I think, you know, having someone to challenge you on some of those things is really key, because we all have blind spots. They, you know, and they never go away. They change, but they never necessarily go away. But no, Lara, I could talk to you about more of this stuff all day, but we are coming to time. It's a very, very exciting time for you guys. We wish you the best of luck.
Thank you.
Thank you for joining us.
Thanks for having us.
Yeah, of course. We appreciate the insight.
Thank you.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium or TikTok. See you next time.